To understand the exceptions in virology, we first have to establish the rule. A standard virus is defined by two mandatory components: a nucleic acid core carrying the genetic payload, entirely enclosed with a protective protein shell known as a capsid. That structural requirement holds true across different viral morphologies. Even when the shape shifts to a helical structure, the core engineering remains identical: genetic code wrapped in a protein delivery vehicle. It is this specific combination of code and casing that allows viruses to infiltrate host cells and trigger massive agricultural losses. We see this with the tobacco mosaic virus, identifiable by the irregular, mottled patches draining the color from the leaf. Plant viruses also force severe structural deformations. The banana bunchy top virus, for instance, results in a choked, clustered growth pattern instead of healthy, spreading fronds. In animal and human pathology, standard viruses drive a massive spectrum of disease. They cause localized, painful surface infections, like the targeted blister clusters seen in a shingles outbreak, and they scale up to cause systemic, historically devastating plagues like smallpox, characterized by severe, widespread pustules across the entire body. For decades, this formed the central dogma of infectious disease. To be a biological threat, an agent required both a genetic blueprint to replicate and a protein delivery vehicle to get inside the host. Then, pathologists began isolating transmissible diseases where the causative agent entirely lacked one of those two basic structural components. We discovered that infection operates at an even smaller, subviral level. Pathogenicity does not require a complete virion. It runs on parallel tracks through two distinct biological anomalies: Viroids and Prions. Structurally, a viroid is stripped down to the absolute minimum. It is exclusively a single, incredibly tiny, circular strand of RNA. There is no protective protein coat and no outer capsid. It is completely naked genetic code. This extreme simplicity introduces an immediate biological limitation. The viroid contains zero genetic instructions to code for its own proteins. To survive, it relies on a purely parasitic reproduction strategy. Once inside a cell, it hijacks the specific proteins already present in the host's machinery, forcing them to bind to its strand and replicate the viroid. Because it lacks a delivery shell to penetrate a host, a viroid cannot force its way in. It relies entirely on physical breaches, slipping through cellular membranes that have already been torn open by mechanical injury or insects. These naked RNA loops are highly specialized. They operate strictly as plant pathogens and have absolutely no effect on human or animal systems. Yet, in plant systems, the macroscopic evidence of a viroid infiltration is incredibly devastating, resulting in severe agricultural deformities. We see this extreme impact in potato spindle tuber disease, a healthy specimen on the left, compared to severely shriveled, elongated potatoes on the right. It causes apple scarskin disease, visually identifiable by the pale, structural scarring radiating outward across the surface of the fruit. And it drives avocado sunblotch, characterized by the deep, irregular yellow fissures cutting directly into the dark green skin. In larger crops, it causes coconut kadang, also known as Bogia syndrome, effectively choking the life out of infected palms and leaving withered, dying fronds. Without a single protective shell, a solitary loop of genetic code proves mechanically sufficient to completely dismantle complex agricultural ecosystems. The prion is the exact structural inverse of the viroid. It is a pathogen constructed entirely of protein. It possesses absolutely zero RNA or DNA. A prion begins as a harmless endogenous structure. It originates as a perfectly normal polypeptide protein, coded directly into the native genes of all mammals. The pathology begins with a geometric error. This normal protein spontaneously misfolds. It snaps into an abnormal, highly rigid structure that acts as a highly contagious template. Unlike viroids, prions completely bypass plant life. They exclusively target animal and human neural organs, destroying the brain. This initiates a specific classification of lethal diseases, known as transmissible spongiform encephalopathies, or TSEs. Once a misfolded prion is introduced, it acts like a domino. It forces adjacent healthy proteins to adopt its abnormal shape, triggering a cascading chain reaction that dissolves neural matter and punches microscopic holes into brain tissue. These MRI scans display evidence of cellular destruction. a healthy hemisphere compared to one undergoing prion-induced collapse. The disease leaves the brain with a spongy texture, seen in the intense white regions and empty voids on the right. This structural collapse occurs across several mammalian species. In sheep, it presents a scrapie, while in livestock, it is documented as mad cow disease. When it crosses the species barrier into humans, it manifests as Creutzfeldt-Jakob disease, or CRU. Because these are highly stable, non-living proteins, they do not die off easily. They spread directly through the consumption of infected neural tissue, via contaminated blood transfusions, or through organ transplants. The prion proves that catastrophic neurological collapse does not require complex genetic instructions. It can be triggered entirely by corrupted protein geometry. To define the boundaries of the subviral paradigm, we place these two agents into a comparative matrix. Their host environments are completely divergent. Viroids operate strictly as plant pathogens, whereas prions act exclusively as mammalian brain pathogens. Their structural compositions are mutually exclusive. A viroid is nothing more than a single circular strand of RNA, while a prion is purely an abnormal polypeptide protein. This reveals the core paradox of subviral agents. The viroid operates with absolutely no protective protein coat, and the prion transmits disease with absolutely no nucleic acid. This introduces a highly counterintuitive reality regarding physical scale. A naked string of biological code actually occupies more physical space, making a viroid structurally larger than a prion protein. The mechanics of infectious disease operate far below the threshold of the standard virion. Complete acellular pathogenesis does not require a fully built delivery system. It operates ruthlessly through a solitary loop of code or a single misfolded shape.